Welcome to another Biota podcast. It's July 30th, 2012, and I have the pleasure of actually spending time with Roy Plotnick. Hello, Roy.
Hello, how are you doing?
Very well, very well. So one of my favourite Biota podcasts was the one associated with whether artificial life could explain the Precambrian period that you and I, and maybe two or three others, did maybe about 5 or 6 years ago now.
It sounds right. It sounds right.
So, in terms of that kind of time frame, I've had an amazing opportunity today to spend time in your lab and get a sense from both the fossils and the simulation what you do.
Right.
But for folks listening in who may not have heard that podcast, can you describe, firstly, what exists in the fossils, and secondly, how your simulations map to what you see in the fossils?
Okay, well, one of the major events in Earth history is the transition from the Precambrian, specifically from the period of time we now call the Ediacaran, to the Cambrian. So we're talking about the period from roughly, let's say, 560 million years ago to about 530 million years ago. If you look at life of the Ediacaran, there are multicellular organisms. Some of them are quite large. There are lots of arguments about what they are, whether they're ancestral to modern day forms. There seem to be one or two that may be of the larger ones that are capable of some limited movement. In terms of the fossil record of movement, most of it seems to be very tiny things feeding on in and under microbial mats. And there is no evidence that anything in that period of time, prior to 543 million years ago, had eyes, had antennae, anything large-scale sensory organs. So the world of the latest Precambrian is very simple in many ways compared to what we see. Large forms living in and on mats. Nothing that would look recognizably that you say, that's an arthropod, or that's an annelid, or that's a bivalve. Things that we think have maybe ancestral to jellyfish, but it's a lot of argument about that. You go up to the Cambrian, you go over 543 million years ago, you start to get organisms with shells, you start to get trace fossils, that's the remains of animal movement, which are no longer simply horizontal but also are vertical. We get indications of the modern faunas coming in by 510 million years ago. You get the Burgess Shale that everybody knows about, the older Chengjian. You've got organisms with eyes, antennae, things like that. So there's a large-scale development of sense organs. So what I'm really interested in is, why do we have this transition between things that were small, very small, mobile, no noticeable sense organs, and very simple traces, tracks and trails, to the much more complex world we find later on, and it's specifically looking at behavior and looking at sensory biology of those periods of time. And this concept I've suggested is that we have a revolution in information, that the world has become spatially, and probably to some extent temporally, more complex as we go over the boundary, as we get more mobile things, as we get larger things, as the sediment starts to get churned out, as biomass gets concentrated in more forms. This makes the world more complex. And organisms that don't have a way of detecting these complexities are at a disadvantage. Now, the way I've been exploring this is by using a compared to what a lot of people do, very simple agent model. The agent model essentially has an organism that examines its nearest neighbor's sites and sees what kind of resources are there, moves into those sites based on which site has the highest amount of resources, consumes what's there. If the nearest neighbor sites don't have a resource, it detects a chemical signal from a further site and moves in that direction. And what I've been saying is about a lot of assumption in the fossil literature that if you find these complex or complicated pathways of movement, that this means that the behavior was complex. But what we actually, the model indicates that spatial heterogeneity, spatial complexity, with very simple behavior can make patterns that look complicated. So my original models I've been working on for a while have been one-dimensional, two-dimensional, just looking at the surface. I've recently expanded it to 3 dimensions, and also adding multiple agents so that they can interact with each other, and seeing what kinds of patterns we can get. I think what we want to do is come up with a minimal explanation for the patterns we see in terms of behavior.
Certainly.
Okay,
Certainly.
so that's in a nutshell.
In a nutshell. So, in terms of what we've done today, and I mean, for the folks listening in, I also had the privilege to show Roy Noble-8 to give him a sense, basically, of my background and also the kind of independent autonomous agents and the notion of space and these kind of things, with the view that certainly what you were describing, and predation is an interesting part of what you were describing as well, because your model doesn't have any form of predation currently.
Right. I want to put it in.
But predation, as you've described it, is things that are either swimming above or around that give some additional element. Can you describe predation in the Ediacaran? I mean, was the predation...
This is, again, a source of some controversy. There's no evidence of bite marks. There's no evidence of tooth marks. There's no evidence of something large consuming something else large. So there's no macroscopic predation seems to be lacking in the Ediacaran. This led the suggestion, some people have called this the Garden of Ediacara as a result, because it was peaceful and quiet. But there are some indications in some small shells in the very latest part of the Ediacaran, just before the Cambrian, of borings. And it's been pointed out by one of my colleagues, if you had something like a jellyfish engulfing something whole, you wouldn't see a record of that. But you don't see teeth. You don't see claws in any of the Ediacaran forms. Those are totally unknown at that point in time. So, again, one of the things that most people think is a large component of the Cambrian radiation. I don't like the idea of that it's some sort of fast. I don't like the implication that it's really fast. While that people, it's over 20 million years, 30 million years, you've got to change, and that's not quite so revolutionary. But the Cambrian radiation, the Cambrian expansion of multicellular life, a lot of that is predation. It may very well be predation driven. We start to get things that have clearly seem to be predation related: eyes, claws, things like that. There's arguments. There's a large relative sense organism called Anomalocaris that seems to be predatory. It has large. It looks like a predaceous mouth. It has large pincers and lacking things in front, claws in front, what you would call large appendages. Has eyes on top of its head and may have been a sit-and-wait predator. We may have had very small predators, things called pedonats, probably right at the beginning of the Cambrian. We find things living up in the water column that may have been predaceous. But clear evidence of macroscopic predation really doesn't seem to come in until the Cambrian, and that's going to change the game. One of the things, again, about predation's expensive.
Certainly.
You have to locate your prey. You have to chase it down. You have to capture it. You have to dismember it. And there has to be enough there at the end to have made the expense of it worthwhile to do so. So, again, one of the questions that comes up is, what is the expense of predation? When does the expense of predation get less than the cost of not predating? When does the cookie you're getting at the end, the piece of pizza you're getting at the end, pay for the energy you've just expended? And again, that's a question of evolving muscles and other tissues. So a bag of jelly doesn't have much to eat on it. I mean, turtles may eat jellyfish, but overall, the richer the organisms are to eat, the more it's worthwhile to be a predator. So a lot of these I think are much involved in. There's a lot of feedbacks going on in the whole system.
So the example of the burrowing snail, or the snail that eats through the shell in order to get the organism, doesn't have to do the dismembering, really.
No.
So in terms of predation, I guess there's a continuum of where the burrowing snail is somewhere in there, but it doesn't have to...
Well, ticids are the moon snails. The ticids are classic predators. They eat... You go to a beach these days, you find shells with a sort of a conical hole in them. And that's produced by these predaceous snails. And that's actually a favorite animal for paleontologists to work on to study predation, because it's so clear. We know who did
definitive,
it. Yes. It's definitive who know who did it.
It's a bullet hole, really, isn't it?
Right, it really is. And there are people who've done studies where they've traced the history of this predation through time. They look at if there's a favorite spot where they predate, or their favorite organisms they predate on. There actually you can find failed examples of predation going on with those. But again, that's there's a lot of a question of how do these animals decide this clam is too big to bore through given the time that I have. They have to watch out for their own. They actually predate on each other. They have to watch out for their own.
So they bore each other.
They can bore each other. You can find, they're at least you know it's safe to eat because it's the same thing you are. But that's one of the classic cases that we know of predation in the record.
Yes.
But that again is you've got the animal has there's a whole field of an ecology of how do an organism decide when to feed on something, when to stop feeding, when to run away and look for another day, things like that. Again, one of the major questions I'm interested in is when did these games really start going? And I'm pretty sure it was pretty early on.
So in terms of artificial life simulating this, there's almost a continuum. I mean, you're starting very much in the kind of your model is Ediacaran.
Or Cambridge, even modern. You could use it for modern day things.
Certainly, but I mean, but in terms of the addition of complexity, in terms of actually. So, can you describe a little bit about what you found in terms of firstly having a very dense, nutritious feeding source that's there, and then moving the feeding source into further spaces? Well, I mean, there's
again if I start with I can switch on and off the organism's ability to detect at a distance. Mm-hmm. So the simplest organism can only detect near field, very close in. So if you imagine something that's just has just taste.
Certainly.
It can stick its tongue out and taste in the vicinity, but can't smell.
Yes.
And so if you do something like that, if resources are distributed continuously, not necessarily homogeneously, but continuously, that's fine. Because you always are going to go to the next spot that has the most food. And you don't really care what's 20 spots over, because eventually you might get there. But if food is patchy, if there's a space between you and the next spot, then once you've eaten what you're in, you have to find that next little rich bit.
Certainly.
And so that's when it becomes necessary to have smell, or vision, or some other way of saying, oh, I need to go that way. And some of my earlier models, I actually played with, for things just randomly goes around, but if you don't have that ability, you just don't get as much as you can out of it. So evolution would select for something with the ability to find something at a distance the moment things became patchy.
Certainly. Just as what you were trying to show in actuality, the artificial life perspective associated with the evolution of senses, and then that is an interesting problem in and of itself too. So, I mean, certainly from our initial discussion 5-6 years ago, the idea of the emergence of intelligence through survival, although clearly at its very most primitive level it's not intelligence, but I mean somewhere through the continuum it becomes intelligence, I found absolutely fascinating, and I think a number of folk who corresponded with me following found that as well. Can you talk a little bit about, I mean, it's one thing to go from something with no eyes, no sense, to something with that. Can you talk about the evolution of these kind of sense organs and how you think it could be modelled from an artificial life perspective?
Well, again, I think, it depends on the sense organ. A lot of things. If you go back to bacteria can sense chemical gradients.
Certainly.
Single-celled protists can sense chemical gradients. So that is not, the actual cellular machinery for detecting chemical gradients is pretty straightforward to make. The great material for gathering information on, once you have multiple parts of the body, I'll go to something like Carl Sims type blocky things. If you have multiple parts of the body and you have something that allows you to determine how much stress is on that, so proprioceptor kind of device, that can very easily be turned into a mechanical receptor for detecting water currents or something like that. That's straightforward to do. In terms of a light, again, there's been, I think this was in one of Dawkins' books, as I recall, there's a famous experiment where they started with a single photoreceptor cell on a membrane, and they were able to evolve it into an eye
Yes.
over time. So it's I don't think it's particularly complicated to get those kinds of things done. I have one of my other lines of research. I've been looking at the hearing in insects, and based on talking to the entomologists, ears in insects evolved independently 17, 18 times. Insects have ears on their legs, like crickets and katydids do. They have ears on their abdomens. They have ears on their wings. They have wings at the base of their thorax. I mean, anywhere you can think you can put an ear, except right on the head proper.
Yes.
You can put. They have developed ears, and they're easy to make. You just take a piece of the cuticle that has a mechanical receptor, a proprioceptor underneath it, with an air sac, trachea, and you thin it.
Yes.
And all of a sudden you've got a drum and you've got something that can detect sound. So, the real issue is I don't think it's all that complicated to develop sensory organs of some kind. What does get complicated is you also need to be able to take the signal from those sensory organs and interpret them.
Certainly, Steve Grand has done a number of things in software and hardware in terms of, for want of a better term, kind of atomizing these elements. And I now maintain an open source version of his work that does this called Symmetry, where exactly as you describe, really bits are connected together and they may end up seeing or hearing or moving, but they all have the same kind of communication interfaces, basically. So that's an interesting... Okay, so let's map Symmetry onto what you're describing. I guess food is primarily survival, is primarily the benefit that you get out of it.
Survival and reproduction.
Yes.
Everything else is secondary.
Certainly. So, okay, so that's very interesting. So you start off, and you allow, I guess, through some kind of random selection, the ability to add one of these elements. So you start off with a full set of elements, but your first one has none of these elements, but it can by that's very interesting. Okay.
Well, there's a lot of indication in molecular biology, and my colleague Dan McShay at Duke would say, in large-scale evolution too, of duplication.
Certainly.
You take something, you duplicate it, you keep the original to keep doing what you originally wanted to do. The other one can change and add complexity, add functionality to what you've got.
Yes.
So duplication of parts I think is one of those essential
So
taking
Symmetry, you have those elements in Symmetry. You would then have a simulated agent that was based from these atomic components, which you would then simulate in free space as opposed to the feeding grid that you have currently.
Right.
And then, over many, many iterations, you could see what came out of that and the change in movement. Now, the predation element is interesting as well. I seem to recall, and I need to double-check the code, but there may be something like teeth or something that consumes as part of these. And that may be an interesting element. The other part that you noted is associated with how, I think we had the discussion maybe associated with the early Cambrian, how predators were detected. And the multiple ways of predator detection. Could you talk a little bit more about that?
Again, you're going to detect predators the same way organisms do today. You can smell them coming.
Yes.
I mean, one of the things that's going on in terms of information, there are far, far more kinds of organisms around in the Cambrian than there were before.
Certainly.
So you need to be able to develop a way of detecting that predator that you can say, that's a predator, it's not a conspecific, it's not a harmless little thing floating by that I don't have to worry about. So the first thing, there is always a smell of some kind. We did experiments, one of my former students, Karen Coy, who's not a professor herself, on worms, nematode worms, where she took a predator, the flatworm, and she basically, you wash it, and you take the fluid from that washed worm, you put it in near to where the nematode is, and the nematode goes, oh my God, there's somebody nearby, and it turns around and changes its foraging pattern. And what's fun about that is that these things have been raised in the laboratory for thousands and thousands and thousands of generations, but they still know that this is something they need to run away from.
Yes.
We've done similar experiments you did with crabs and with snails. In that case, the predation clue was generally you take one of their congeners and you destroy it, you kill it, and then you put the dead thing in, and they say, oh, somebody's eaten one of my somebody in my species, I should hide, I should run away.
Yes.
So chemical detection of predation is one of the most fundamental ways you do it. Either the predator itself or some sort of fluid from somebody who died of your species, and you should say, okay, maybe there's somebody nearby I need to be careful of. And of course, there's the usual visual kinds of methodology too, that you want to be able to do that. All this again is the major issue is you got to be able to process it. You got to be able to do something that says that's not just somebody else, but that's that one out there, and it becomes the more signals you have to process becomes, as you know, more exponential growth in the amount of processing that's needed to do, which has a cost of its own.
Certainly. So artificial life as it could be.
Right.
This is something that I found very interesting spending a day with you today, is that there's still many unknowns in what you're looking at. And what artificial life could provide is actually potentially a multiplicity of solutions that could then map down onto what you're specifically looking for. Can you talk a little bit about that?
The question that I wonder about, and this one of the reasons I started in this whole field is that Steve Gould wrote his book Wonderful Life. That inspired Bruce Damer and others to put together the first digital Biota conference to sort of say, is the growth of worlds of artificial life, artificial intelligence, worlds of the computer itself, the internet, all that, just starting out in those days, is the Cambrian explosion, again, I hate the word, explosion,
certainly, yes,
similar to that. So the question, the issue becomes is that's essentially how I got into that. Now, Gould, in his Wonderful Life, raised the issue of what is called contingency. And he often held up the example of he used the phrase, if you ran the tape of life again, what would life be like? So if you were to go back to the Cambrian, find the early ancestral vertebrate and make it go extinct as opposed to all the other things that went extinct, would we be around? And his attitude, his express attitude was, life is highly contingent upon individual things. On the other hand, Simon Conway Morris, who was very heavily written about in Gould's book, has written several things where he's strongly disagreed. He feels that there's an inevitability to much of life, an inevitability to even to intelligence, that intelligence would evolve again. So he disagreed. We recognize contingency happens. I mean, if the asteroid hadn't hit us, we might not be here. Certainly, the dinosaurs may or may not have gone extinct. Big argument. Don't want to go into that. But there are certainly contingent elements in history. But the big debate, the big philosophical debate, I think we always had in paleontology and continue to have, is what extent is the history of life contingent, based on random happenings, random events, versus purely deterministic. Eventually something will happen.
Yes.
And I tend to say that there are certain things, because of the nature of chemistry and physics, that are going to have to be, because, and this is where artificial life comes, because of the nature of complex systems that have to be. So that if you were to go to another planet, if you were to run the tape of life again, yes, we might not have us, but you would still have organisms that were filter feeders.
Certainly.
You would have organisms that were deposit feeders.
Yes.
I think ecology, and players may be different, but I suspect that if you were an ecologist and you went to another planet, you could do ecology, and there wouldn't be anything that would be fundamentally different. In order to have evolution, you have to have reproduction with duplication, evolution, and mutation. The basic processes would be recognizable. So I think artificial life gives us a way to say, what about the evolution of life is by chance, by accident, and what is something that is just a function of the way systems have to behave, complex, interacting systems. And I suspect that, again, my suspicion is that we will find, again, aliens won't look like, as was it Star Trek, they call it forehead aliens?
Yes.
Yes. Well, it's just humans with a different forehead and on top of their head.
Yes.
But we'd still find life on other planets if we found it, as long as it's water. recognizable to a broad sense, even if it's not DNA, even if it's not the other things.
Certainly.
So until we get to those other planets, I think A-Life lets us play with those ideas.
Yes, and I mean certainly something which today has impacted on as well for me was the teeming nature of it. The fact that, as you say, fossils aren't rare.
No, they're
But
not.
there are so many, and the trace fossils in terms of the tracks and all these kind of things, it, visually it gives you a great sense of just the sheer numbers involved, which is ultimately a parallel computation fundamentally on some.
Right.
And so yes, in these ways, although the asteroid is an interesting one, but contingency certainly came up at the last A-Life conference that I was.
No, this is, and
I think it's a.
I think I raised this issue in the last time I spoke to you.
Mm-hmm.
But one of the things that I had trouble with some of the A-Life programs I had is they didn't have the disaster.
Yes.
Things would go on and evolve along, but you wouldn't have a sense where we're just going to randomly knock out half the things that are in there, or 90% like at the end of the Permian.
Mm-hmm.
What if we were to sort of really hit the system hard? Computers crash,
Certainly. Yeah.
We're not going to crash the simulation, but have something in the simulation that says, let's not continue as it was. Let's do something that's unpredictable in the event of it and really hit it and see what happens as it comes back up again.
Mm-hmm.
So that aspect of contingency, I think, is something that I would love to see thought about in A-Life models.
Certainly. And I mean, as we were discussing last night, things like temperature and change in the atmosphere and these kind of things could be very interesting to add to artificial light simulations as well.
Long term, and again, it's a computational problem, as you know, but physical environments themselves shift. Instead of having maybe I'm a little, maybe too much, a little loop, but having a moving target.
Certainly.
In terms of the physical environment would be a very interesting game to play.
Certainly.
Because, again, we see this in ecological systems. Ecological systems, it's the assumption is if the environment is stable, you evolve to a climax state, and then everything will never change.
Yes.
But it's very clear that ecological systems, even in the absence of what humans have done, are always being reset. In fact, that disturbance is a major component of actually long-term system stability in natural systems. That you go on, and there's a classic thing, if you have two species that are competitors with each other, the only way you usually can get them to survive is if you have something either predating on both of them, or there's enough disturbance to keep them together. If you don't have that kind of external disturbance in the system, then you may be getting things that are not realistic.
Certainly. So one of the other points that I took away from the A-Life conference was the lack or distrust of complexity. It was one of these strange things, because, I mean, doing what I do with Noble Ape away from the academic A-Life community, at least, I love the ability to take multiple simulations and put them together and see what comes out of that, turn one of them off, these kind of things. But the Artificial Life Conference really returned, I think, to a more traditional model. It was actually really quite curious. And the thing that I liked was the special issue that you brought out that actually acknowledged, multiple effects that fed back into movement, which was exactly Noble Ape associated with external diversity and all these kind of... Can you talk a little bit about that and how it maps onto your work?
Well, this was a special issue of the PNAS, Proceedings National Academy of Sciences, in 2008, I think it is, on movement ecology.
Certainly.
And Ryan Nathan and his co-workers have put together a number of papers trying to come up with a way of synthesizing how ecologists look at movement. There is a huge literature. I mean, I think one of the papers in the issue summarized 10 years worth of papers that discuss movement in plants and animals and protists, and there were like 25,000 papers or some ungodly amount like that. And so they said, and the problem is that everybody uses a different language to describe movement. Everybody uses a different approach to thinking about movement. And so what they propose is what they call a movement ecology paradigm. And the movement ecology paradigm essentially says you can really look at movement, and I'm going to simplify greatly here, as a set of things. First of all, you have the where to move. So an organism, let's say the navigation capacity. How does an organism decide where it should move to? And this is going to be a function of its sensory capabilities and its neurological capabilities of processing that information. Then the second part of it is essentially the locomotory abilities, the how to move. I might see something I'd like to eat swimming above my head, but if I can't swim, I can't get there. So there are certain biomechanical constraints that organisms have. Organisms have different abilities of being able to move. So I can sense something's up there, but which I might either want to move to, if it's a mate or a food item, or away from if it's a predator. But I also have the ability to move towards or away. The third element of this is the internal state of the organism. And so the internal state of the organism is, I'm hungry, I'm satiated, I want to mate, we've mated, I'm smoking a cigarette, those kinds of internal states, which are really hard to get in either modern or fossil organisms. But that is the decision of the why to move, as Rand put it. So you have those sort of three things, and you can put pretty much all organismal things into sort of that kind of the where to move, how to move, why to move, and their interactions with each other. Those are all embedded further into the question of the external environment. So that could be, again, the physical environment or the biotic environment. For an organism, again, a potential mate walking by is going to be external environment. Mm-hmm. That may, again, if your internal status says it's time to mate, then you go, oh, there she is, and you go towards it, and you move towards it. But if you smell it, it's a male, and you're a male, well, you may be not that interested, perhaps. So this isn't, external environment is sort of the context in which all this occurs. So you put all these together, these all interact with each other. And they interact over time, and interact over space. But the end result is the organism either stays in one place, which is one kind of movement phase, or the organism moves. The end result is a movement path. Mm-hmm. So the resulting movement path, and we studied the movement path in modern organisms by radio collars or by photographing them, and there's a tremendous amount of research on things like that, is the idea that this movement path is what we end up studying, and it is a result of all these features. And what I'm trying to do is say, I've just published a paper on this, is saying, well, in actuality, this can be applied to fossil organisms as well. They had the same questions of the where to move, the why, how to move, the why to move, mm-hmm. and then what we have a lot of trace fossils. That's the movement path. So I've tried to bring that conceptual development into how we look at trace fossils, which I hope is going to make a change of how people talk about trace fossils. I'm going to a meeting in a few weeks. And hopefully I won't, of trace fossil specialists, and hopefully I won't get yelled at. Or if I will, so what?
But I think it's also an important perspective for particularly artificial life developers that are looking at simulated agents, and mm-hmm. maybe even simulated agents that would be beneficial to biologists that are studying these kind of things, to actually embody that in their simulations in some way.
Yeah, the framework can certainly be brought into A life. I don't see why not.
Certainly. And I think, as you've seen with Noble 8, a number of those components are in there already, and I kind of put them in implicitly, associated with just creating layered simulations. Mm-hmm. But it's very interesting that is being explicitly recognised as a mechanism, and I think certainly that kind of feedback into perhaps the academic artificial life community is very powerful as well. And talking more in terms of the practical side of things, certainly something that caught me through the A-Life conference was that I have a squandered luxury in some regard in terms of the folks that come through and like developing a Noble Ape and ask for, mm-hmm. Projects and these kind of things. And certainly I found at the conference maybe half a dozen papers that immediately mapped back to what I was doing in Noble Ape that would be relatively easy.
Right.
You are very much, in what you do, you develop your own software, mm-hmm. you work very much, but in terms of ideas of open source and these kind of things, you're married to your language to a certain extent.
Yes.
But there must be a kind of strength of numbers where potentially if you got developers that were working on it or it was part of an open source program or something like this, you could start seeing what you wanted to see without the kind of maintenance that you have currently. Can you talk
this
a
is
little
a bit of background on this. The first computer language I ever learned was Fortran. That's back in high school, more years ago than I want to talk about. I've had Fortran programming classes. My advisor had programming in Fortran, so I've always programmed in Fortran.
Yes.
I'm currently using Fortran 95 with a Windows interface called Winteractor, and I'm very comfortable programming in Fortran. And I've looked at going to C, and I just didn't feel the comfort level was there for me to make that transition to programming in something else. And as I think Brian Hayes once put it, it's not it's not always the best language, it's the language you know best. That said, I have absolutely, no objection if somebody wants to look at what I've done and take the basic conceptual concepts and do it in a different language that might be better for me, and maybe at the same time help me learn that language. I'm not married to Fortran, it's just where my comfort level is. For me, the major issue is I have to know what the program is doing. I have to understand the guts of it, and it has to be able to still address the questions that I'm interested in as a scientist. Part of this is I had a program at one point working with sort of the ancestral program to my trace fossil one called CAPS. And CAPS was a cellular automata-based program to look at seed dispersal and disturbance. Working with my colleague Bob Gardner in Maryland on this one. He did almost all the programming again in Fortran because that's what he's comfortable with also. And at one point we hired a young programmer who was doing it in one of the C-based languages, either C or C++. And he did things, and I just said, I don't understand what you did. I don't know why you do this. So, well, this is the best way to do it. And I said, well, but
So it is an interesting problem, and certainly, for background for folks listening in, the fractal model that Roy uses for the placement of food is very similar to the fractal model that I use for landscape generation with Barbalet. And I think probably about if there was more commentary in there, probably 90% of the code you were relatively comfortable with. It was a few optimizations that I'd done that, in reflection, to have people such as yourself use the code, I would perfectly easily remove those optimizations.
Right.
Because my view is that humans are more valuable than processing power on some fundamental level. So I can see, what you present with your Fortran, and these kind of things, is a relatively small step away from C if the C is presented to you in an equivalent.
That's correct.
Kind of way.
Yeah.
And I think that's interesting that, and certainly this is something I found working with the MSU folk as well, is that there are certain things that I put in as shorthand in Barbalet, which really shouldn't be in there for, long-term academic use.
Right.
And I think it's interesting from an engineering perspective to actually identify if it's going to be used by scientists that are particular, that it needs to be written in a very particular way.
It needs to be, again, anytime you do the whole question of program validation verification, yes, you need to annotate it, but it needs to be more just than what the code is doing, but it's also what questions are being addressed. And I think that's the fundamental tension we always have between programmers and the scientists using it. Scientists, I've had been told on one of occasions that we shouldn't do programming. This issue has come up occasionally, that we should leave it to the professionals. And my attitude is, but I know what questions I want to ask, and I want to be able to write code that lets me do that.
Yes.
Now, it may not be the best code, but I know what it's asking in this case. And I may not be the sickest interface, but I know what it shows me the answers I'm hoping to get to on these things.
So, Roy, in terms of broader things, we had an initial discussion last night about my perspective on the A-Life conference, because I think you felt not necessarily out of touch, but that you just didn't have a connection with what was going on currently.
I certainly don't. I occasionally listen to the podcast, and I check it. If a paper comes out that I see on robotics or A-Life that I find interesting, I will look at it. I know some things have come out in the last couple of years and sort of catch my eye. But I haven't had much active contact with that community, I would say, since the last Biota conferences were held. And that's maybe partly my fault, partly...
I think it's gone in a variety of different directions, so it's really quite a difficult community to track.
Yeah.
And aside from, as you described, the kind of passive way. But in terms of what you're working on, I think you've given a very clear definition of what you'd like to see in an assistive sense, at least. But in terms of the broader community, I mean, for the past week, I guess, I've been meeting with scientists, and it's been a real luxury in time to get a sense of what you are working on, and also what I can do to assist with what you're working on.
Right.
With the view that, certainly with my work with Barbalet and Biota, it is fundamentally a volunteer effort.
Right.
And it's an interesting problem as well, because certainly in terms of industry and industry support of these kind of things, there are ways in which industry can pick up these programs and work with them too. It would give support here as well. So in terms of the broader themes, artificial life community, these kind of things, what more would you like to see in the next decade?
Well, it's really hard to say. I would love to, I'd be more than interested to talk to people, and I guess this is one form we're doing it right now, about what real life actually is happening. Artificial life is interesting to me if it sheds light on, I don't know, what you call real life. I mean, biological life. If it sheds light on biological life and the kinds of things that are going on with biological life. My concern is sort of if a life becomes its own self-generating, we're just going to a thing where it's only interested in a life research and a life ideas. I was talking with you about this earlier. There was sort of this little area of physics that came out of Pierre Bach developed things on self-organized criticality models for evolution. And this became a cottage industry in the physics journals. There was just continually papers coming out. A lot of them I got sent to review, where they were sort of looking at, self-organized criticality about evolution and extinctions, this and that. But they never went back clearly to the original literature and never talked to paleontologists or evolutionary biologists about what they were doing. I'm a great believer. My career is multidisciplinary. I'm probably more than most of anybody else in my field. I go way out of paleontology, like into A-life and the fractals and things like that. I would welcome if somebody's saying, gee, here's this model I'm doing and has something to say about the Cambrian explosion, if they actually came and talked to us about what we know about the Cambrian explosion or the extinction of the dinosaurs or patterns in life, and what's the current thinking, what's the current literature, what do we say about it? So I think it's, in general, I think most paleontologists would love to. We all love to talk about what we do, because otherwise, why do it? But I think it's. I'm interested to hear what people are coming up with, but I'd like to also know that come and talk to us too.
Certainly
about what we're doing.
Certainly. So the outreach goes to the community, basically.
Yeah,
it's the responsibility of individuals to make contact and do this kind of.
I'm a tremendous believer. This paper I just wrote, I just had come out on the trace fossils. Let me give you. This is an example of this kind of intercommunity thing. Trace fossils, for you may not know, are fossilized tracks and trails and burrows and borings and all the things that animals or plants produce through their life activities. So it is the fossils of behavior, if you want to think about it, or fossils. It's when something does a behavior that interacts with the substrate and leaves a trace behind. Not all behaviors leave a trace. And there's a large community in paleontology and in sedimentary geology of people who work with trace fossils as environmental indicators and things such as that. There have been writings about these things. They say, it's all, trace fossils are evidence of behavior. But if you go into the literature of trace fossils, traditional literature of trace fossils, and you say, okay, well, these are people talking about behavior. What models, what concepts from the animal behavior literature do they bring in? It's not there. There is scarcely a mention of any of even the most fundamental Tinbergen type 4 questions of animal behavior in the trace fossil literature that doesn't exist. At the same time, if you go into the trace fossil, the papers of the books about animal behavior, the textbooks of animal behavior, they might say, well, we know we have dinosaurs had nests. That may be a little mentioned. But the idea that the behavior has evolved over 550 million years, or 4 billion, or since the origin of life, is not in the mindset of behavioral biologists. It isn't in their books. It isn't in their papers. They talk about evolution. It's very short term. And so this paper I wrote was sort of like, hey, guys, you got to talk to each other. Mm-hmm. And I'm a tremendous believer in we don't want to reinvent wheels, that people in different fields have real, maybe different perspectives on things, and talking to each other, and in the end result, helps everybody. So, I've been talking to people outside of paleontology for years. Mm-hmm. And I encourage it for everybody else to talk to people. Very good.
All right, it's been an absolute pleasure spending today with you and being able to stay with you. And thank you very much for the opportunity to be recorded on this Biota podcast.
Okay.
And hopefully it won't take 5 years before our next recording. So, because, yes, it's always insightful. So thank you very much.
Thank you. All right.